Welcome back to part B of lecture 2, motor system. So we are still discussing reflexes. So I hope by now you understand how you can classify reflexes. So that we discussed in part A of lecture 2 of neurophysiology or neuroscience. So in this lecture, or part B of lecture 2, we're going to look at muscle, spindles, and Golgi tendon organs. And then after this, we'll look at a typical example of reflex mechanism, just a few reflexes, the mechanisms that are involved there in a reflex action. And also muscle tone, we'll discuss muscle tone. Then towards the end of this part B, we'll discuss the general properties of reflexes. So you need to understand those general properties of reflexes. So without wasting time, let's start by looking at muscle spindles and Golgi tendon organs. And these, they're involved in stretch reflexes and inverse stretch reflexes, which we also discussed in part A of motor system lecture 2. Okay, so let's proceed. So this picture here is just showing us details of the spindle fibers. So you can see this muscle spindle, we have innervation to the muscle spindle, then we also have sensory innervation and also motor innervation to the muscle spindle. So you can see the sensory nerve fibers that are innervating the muscle spindle. So when you're looking at the muscle fibers, we have two different types of muscle fibers. We have the muscle fibers that are purely involved in contraction, which are called the extrafusal fibers. So on the peripheral, we have these extrafusal fibers that contain a lot of actin and myosin, and these are the ones that are involved in contraction of the muscle. Then at the center of the muscles, we have special types of muscle fibers. These muscle fibers, they don't necessarily attach to the tendons and then to the bones. They're just attaching to the connective tissue within the muscle itself. So you have those connective tissue, which is called fascia or aponeurosis, that are found within the muscle. This is where they are attaching. But the extrafusal fibers, they attach to the tendon, which the tendon is attaching to the bone for the movement of the joint, for you to produce a movement or whatever you want to do. The intrafusal fibers, they are called intrafusal fibers because they are covered by a sheath. So you can see a sheath of this connective tissue that is covering the intrafusal fibers, separating the intrafusal fibers from the extrafusal fibers. So the intrafusal fibers, they're not mainly involved in contraction. So the middle part of the intrafusal fibers doesn't contract at all, but the peripheral, towards the end of the intrafusal fibers, you can find few actin and myosin that are also involved in contraction. And that is also involved in muscle tone mechanism that we'll discuss towards the end. So you can see here that the intrafusal fibers, they have these sensory nerves that are innervating them. So you have the type 1A, and you know to say the type 1A is an example of A alpha fibers, which are very fast and they are myelinated. So you can see the diameter that it's big here. It's about 17 micrometers. Then we have the type 2. The type 2 are the same as the type A beta sensory nerve fibers. So these are the ones that are innervating the intrafusal fibers. So the type 1A, they are called the primary endings. So these are sensory endings. Then type 2, these are called the secondary endings. So you have two innervation to the intrafusal. Then you can also appreciate the motor nerves that are innervating these muscle fibers. So you can see you have the alpha and gamma. So these are called alpha gamma loops. So you understand the alpha and gamma loops. So the muscles, the fibers that are innervating the intrafusal mainly are the gamma. The alpha motor neurons, innervate the extrafusal fibers. So these, they are very fast. You can see even the diameter, it's bigger as compared to the other motor neuron, like the gamma motor neuron. So these intrafusal fibers, you have two different types of sensory part of the intrafusal. We have the nuclear bag and the nuclear chain. So the nuclear bag intrafusal muscle fibers, it contain like a bag with a lot of nuclei in them. That's why they're called nuclear bag intrafusal fibers. Then we also have a nuclear chain. A nuclear chain is whereby you more like have a chain of nuclei within the muscle fiber. So this is what I'm talking about. We have the nuclear bag fiber, the intrafusal fiber, then you have the nuclear chain fiber. So when you check inside of these fiber, in the nuclear bag, you have a bag with a lot of nuclei there. Then the nuclear chain is just like a chain of nuclei in a fiber. So they are innervated. So you can see, you can appreciate the, the group 1A fiber that are innervating. These are primary afferent. Then we have the group 2 fiber. These are secondary efferent fibers. So these are efferent and these are afferent. So these are sensory and these are motor in function. So these are the nuclear bag fiber and the nuclear chain. Then you can also appreciate the dynamic gamma fibers and the static gamma fibers. So the dynamic gamma fibers are mainly innervating the nuclear bag, and the static, they are innervating the nuclear bag and the nuclear chain. Okay, so just the same example here of a spindle fiber. So in here, you can appreciate the primary secondary endings, which is a type 1A fiber. Then you have the type 2 fibers. So these are secondary sensory endings, the type 2 fibers. So type 1 and type 2, type 1A and type 2. Then these, they are transmitting action potentials from the nuclear bag and the nuclear chain once they are stimulated to the spinal cord, and then they are going to form a synapse with the motor neuron that will now transmit the motor information to the extrafusal fibers and also to the intrafusal fibers. So you can see the gamma efferent motor fiber to the spindle. So these are the ones that will cause the contraction of the terminal ends of the nuclear chain and the nuclear bag muscle fibers. These are the intrafusal. At the center here, you don't have much of actin and myosin, so you don't have much of contraction taking place here. But as you are moving towards the both ends, you can find a bit of actin and myosin that is involved in contraction. But the actual muscles or muscle fibers that are involved in contraction are called the extrafusal fibers. Okay. Then from the tendon, you can appreciate the Golgi tendon organ here. We have the sensory fiber that is also transmitting action potentials to the spinal cord. Then from there, it will form a synapse with the alpha motor neurons that are innervating specific muscles, or it can inhibit other muscles. Okay. So these are Golgi tendon organ. So you have 1B fiber. So type 1B fiber are the ones that are innervating the Golgi tendon organ. So type 1B, these are also A alpha, so they are very fast. So these are the ones that are involved in proprioception. So this is called a spindle, muscle spindle and Golgi tendon organs. So how they work? So if there is stretching of the muscle, it's going to stretch the intrafusal fibers that will stimulate the sensory fibers. So the sensory fibers are going to fire an action potential. So you can see you have type 1A fibers, which is sensory or the primary nerve endings. Once they are stimulated because of stretching, they will fire an action potential that will transmit action potential to the spinal cord. Then it will form a synapse with the motor neurons. So there are two motor neurons here, the alpha and the gamma. The alpha is the one that is innervating the extrafusal fibers, and the gamma is innervating the intrafusal fibers. So it's going to stimulate both motor neurons that will bring about contraction of the extrafusal and the endings of the intrafusal fibers that will bring about contraction in the muscles once there is stretching. So you can see the stimulus here is the stretching, then to bring about contraction. So these are protective reflex that will protect the muscle. So the spindle fibers, they're involved in stretch reflex, and the Golgi tendon, they are involved in inverse of the stretch reflex. So if you don't understand that, you can just go back to part A of the lecture, you'll be able to understand that. Typical example of a reflex mechanism, so a withdrawal reflex. So for instance, if there is a noxious stimulus that will cause pain, so the stimulus is the heat from the hot object, then once there is heat, the nociceptors will be stimulated. So the stimulus is detected by the receptors in the dermis of the skin. Since these sensory cells respond to temperature, they are called thermoreceptors. So the thermoreceptors will be stimulated in the dermis of the skin. Then they will generate a receptor potential that will result into an action potential, and this action potential will be transmitted via the sensory nerve towards the spinal cord. Then the thermoreceptors initiate nerve impulse that pass to the spinal cord along the sensory neuron. Then from there, you have the integratory center, which is the interneuron within the gray matter of the spinal cord that is going to process the information. So the sensory neuron enters the spinal cord via the dorsal branch of the spinal nerve and form a synapse with a relay neuron inside the gray matter. So the relay neuron is the other word for interneuron. So the short relay neuron forms a synapse with a motor neuron that leaves the spinal cord via the ventral root of the spinal cord. So the sensory neuron is entering via the dorsal root, but the motor neuron is leaving via the ventral root of the spinal cord. Then this is the motor neuron, the alpha motor neuron, that is innervating the extrafusal fibers. Now the motor neuron carries the impulse to an effector, in this case muscles in the arm, which contract to withdraw the hand from the hot object. This action is known as a response. So you're going to move from the hot object. So this is just the mechanism of a withdrawal reflex, the pathway. So this is also called the reflex arc. Okay, what about the spindle fibers? How can you activate them? So we said if there's stretching of the muscle, they're going to stretch the spindle, muscle spindles, and then the sensory information will move to the spinal cord, then back to the muscles for that muscle to contract. So innervation of the skeletal muscle, the arm is commanded to a particular position when it's not stretched. So now a weight is applied to the arm. So if a weight is applied to the arm here, you can see that there is stretching in the muscles that will result into stimulation of the spindle fibers. Then the sensory information will move to the spinal cord. Then it will stimulate another action potential that will move via the motor neuron to stimulate the muscles to contract. Then you can see the contraction of the muscles. So this is a protective reflex that will prevent the muscle to tear or to break as a result of overstretching. Then the opposite is the inverse or the stretch reflex. In this diagram, you can also see what is happening. If you are holding a cup in a particular position and then you start filling the cup with water or tea, whatever it is, you find that this cup will be becoming heavy. That will start pulling the hand down. Then that will result into stretching of the biceps. Once the biceps are stretched, then the sensory information will be generated because of the muscle spindles, and it will stimulate the motor neurons to stimulate the muscles to contract so that you are able to support the hand so that you don't drop the cup. So this is just an example of a stretch reflex as well. Enough of the reflexes, let's move to muscle tone. So this could be a tricky question if you didn't really study. So the question can just come say, define a muscle tone. What is muscle tone? What do you understand by muscle tone? Or briefly write about muscle tone. So what information can you write here with regard to the muscle tone? So the word tonus was first used to designate the state of contraction of resting muscle by Muller in 1838. So the word tone comes from the word tonus that was described by Muller in 1838. Vapian defined tone as a state of permanent muscular tension. So muscle tone is a state of permanent muscular tension. So there's tension in the muscle, even when the muscle is not contracting. So that is called muscle tone. In other way, muscle tone is usually described as the resistance of the limb to passive movement. So if you're doing passive movement, maybe in a patient, sometimes you can have resistance to those passive movement. So that resistance is due to muscle tone. So the physiology of muscle tone, control of muscle tone, there are two control areas of the muscle tone. We have the spinal control of muscle tone and supraspinal control. So spinal control is at the level of the spinal cord. The supraspinal control is above. So you're talking of the higher centers of the brain that can also control muscle tone. So let's start by discussing the spinal control of muscle tone. So the spinal control of muscle tone, the stretch reflex of sharing tone is the basic mechanism of tonic activity. So the same stretch reflex is behind the muscle tone or the mechanism of muscle tone activity. I'll explain. So the muscle spindle and alpha and gamma motor neurons are mainly implicated in this muscle tone. So you have the alpha gamma loops that are communicating with the muscle spindle. Remember, the alpha and gamma, these are motor neurons, but you also have the sensory neurons that are stimulating the alpha and gamma motor neurons. So they are afferent and efferent pathways that are responsible for muscle tone. So the sensory, which are called the afferent pathways, you have type 1A from the nuclear bag fiber that passes via the dorsal horn to synapse with the alpha motor neuron. So you can see the type 1A, which is the sensory, which is coming from the primary sensory endings within the intrafusal fibers. This is the sensory that is moving via the dorsal root. Then within the dorsal horn is going to form a synapse with a motor neuron. So you can see this is the alpha motor neuron. Then we have type 2. So the type 2 is also sensory. So it's coming from the endings of the intrafusal fibers or peripheral of the intrafusal fibers, if you want. So from muscle spindle synapse with the interneuron. So this one is forming a synapse with the interneuron, then a motor neuron. That's why these endings are called secondary and not primary, because they're going to form a synapse with an interneuron before it forms a synapse with a motor neuron. Then you have type 1B, which are coming from the Golgi tendon organs. So from the Golgi tendon organs ends in nucleus dorsalis and synapse with an interneuron. So you can see these that are coming from the Golgi tendon in tendon will form a synapse with the interneuron or the relay neuron that is now communicating with the other neurons and other interneurons, which are called the Renshaw cells. Then the Renshaw cells can communicate with the motor neuron. Then the efferent pathway, these are motor. So you have the alpha gamma loops, alpha motor neuron to the extrafusal fibers and the gamma motor neuron to the intrafusal fibers. That will cause contraction of the intrafusal. So what's the actual mechanism behind the muscle tone? What is responsible for those partial muscle contraction when the muscle are relaxed? Even when the muscle are relaxed, you can feel that there's a tone there. So what is responsible for that partial contractility of the muscles that will bring about muscle tone? So here is the tone mechanism. So we start from the gamma motor neuron activity. Remember, you have two motor neurons, one innervating the intrafusal, the other one, the extrafusal, that are involved in contraction. So the one that will bring about muscle tone is the stimulation of the alpha motor neurons. So within the central nervous system, you have the firing of the gamma, not the alpha. The gamma motor neurons is going to fire and it's going to stimulate the intrafusal fibers. Remember, on these ends, or towards the end of the intrafusal fiber, you have actin and myosin that will bring about contraction. So if the gamma is firing an action potential, this part of the intrafusal, they're going to be stimulated. That will bring about contraction. So as they are contracting, the ends are shortening, but they will create a stretch at the center of the intrafusal. So you find that that is going to stretch. So the alpha motor neurons and the gamma motor neurons activity causes the intrafusal fiber to contract. So as the intrafusal fiber are contracting, these stretches the primary sensory ending, thus increasing afferent discharge. So as the endings of the intrafusal are contracting, to bring about stretching of the sensory nerve endings. So this will bring about firing of the sensory nerve endings. Then you find that this type 1A is going to fire an action potential that is going to stimulate the motor neuron, the alpha motor neuron. Then the alpha motor neuron, once it fires an action potential, is going to stimulate the extrafusal that will bring about some contraction of the extrafusal that is responsible for muscle tone. So once there is contraction, it will result into depolarization of the alpha motor neuron supplying the extrafusal muscle, thereby increasing the muscle tone. So this is what is going to increase the muscle tone. So the mechanism of muscle tone, you need to start by mentioning that there is stimulation of the gamma motor neuron activity that will bring about contraction of the intrafusal, stretching the primary nerve endings that will increase the firing, so they'll be stimulated. And then the type 1A fiber, sensory fiber, that will fire action potential that will stimulate the alpha motor neurons in the spinal cord. Then the alpha motor neurons will result into stimulation of the extrafusal muscle fibers. Contractility of extrafusal muscle fibers is responsible for muscle tone. The control, we have we started by discussing the spinal control of muscle tone, mainly looking at the shearing tone, reflex mechanism. Then we also have the supraspinal control. The supra, you're talking of the brain itself. So the efferent fibers to the muscle fiber, muscle spindles. So the efferent fibers to the muscle spindles, the gamma motor neurons receive input from higher centers. So there is input that is coming from the higher center, which is the brain itself. And this input could be facilitatory fibers or inhibitory fibers. So this gamma motor neuron that is initiating the muscle tone activity or mechanism can be stimulated or can be inhibited. So it can be facilitated by the facilitatory fibers, or it can be inhibited by the inhibitory fibers that are descending from the higher center. Mainly, these fibers are originating from the reticular formation. So you have these fibers, I hope you can see in this diagram, you have the reticular formation that is found within the pons. So within the pons, you have excitatory reticular formation of the pons. So this excitatory reticular formation of the pons via the reticulospinal tract of fibers is excitatory. So it's going to stimulate. So you can see the one shown in green here. This is the gamma motor neuron. So if it can be stimulated, then you know to say that it's going to stimulate the contraction of the intrafusal fibers. You can see this one in green. Once it has been stimulated, it will bring about contraction of the intrafusal fibers. Once the intrafusal fibers are contracting, they will stimulate the type 1A sensory neuron that will stimulate the alpha motor neuron that is innervating that muscle, that will bring about contraction of that muscle. And then you have muscle tone. Then you can have inhibitory fibers. These inhibitory fibers, they are coming from the medulla oblongata. So inhibitory reticular formation of the medulla oblongata, it's inhibitory. It's going to inhibit the gamma motor neurons, then the tone will decrease. So there is balancing of contractility and relaxation. So you find that other fibers are contracting, other fibers are relaxing. So you can't say that the muscle is contracting, you can't say that the muscle is relaxing. So there's just muscle tone, which is a partial contractility of the muscle. Okay, so with that information, let's just discuss the general properties of reflexes. So this is just the last part of lecture 2 in motor system. Okay, so let's start with the general properties of reflexes. So there are about 7 properties of reflexes that you need to know. So the first one is adequate stimulus and stereotyped response, reflex time and central delay, recruitment, irradiation of stimulus, after discharge, final common pathway, then you have central, excitatory, and inhibitory state, habituation, and sensitization. So these are the 7 general properties of reflexes. Just adding more information to each property so that you are able to understand what we mean by adequate stimulus and stereotyped response. So adequate stimulus and stereotyped response. So initiation of a reflex is because of a receptor. So you need to know that a receptor is involved in a reflex action. So you need adequate stimulus that is going to stimulate a receptor potential. Otherwise, if you have sub-threshold stimulus, it's not going to stimulate a threshold potential. Without a threshold potential, you're not going to have an action potential in the sensory nerve fiber. Without that, no reflex act. You're not going to get a response. So this also brings us to all or none law of action potentials. It's either you have an adequate stimulus or you have a sub-threshold stimulus that is not going to initiate anything, no action potentials. So that's the adequate stimulus that you need to stimulate the receptor. And the response is always the same and fixed, meaning that once you have an adequate stimulus, you're going to get a response that is always the same and fixed. So it's stereotyped. So stereotyped, it means that you know exactly the response that you're going to get when you apply the stimulus. Like for a knee jerk, you know to say if you stretch the patellar ligament, you're going to have a knee jerk as a response. If you introduce a stimulus on the saw, you know to say you're supposed to get a negative Babinski. But if you get a positive, then that will interpret something else with regard to the function of the corticospinal tract. So you know the stimulus and then you know the response that you're going to get. That is stereotyped. For instance, you have multiple linear tactile stimuli like coating of a flea. It's going to initiate a scratching reflex. So anything that is crawling on the skin, producing that linear tactile stimuli, it will result into scratching. So you're going to scratch. For instance, if something is moving in your scalp, so if it's a linear or a fluid that is moving, it's going to stimulate you to scratch. So this scratching is a stereotype response that you get when you have a multiple linear tactile stimuli that is moving on your skin. You're going to scratch. Or maybe something that is itchy, you're going to scratch. Then the second property, reflex time and central delay. Time elapsed between application of the stimulus and the response elicited. That is due to conduction time and synaptic delay in the central nervous system. So the central delay is the time taken for impulse to pass through the spinal cord and is related to the number of synapses, how many synapses are there. So if it's monosynaptic, it won't take much time. If it's asynaptic, then it's even shorter. Polysynaptic, it will take much time. Bisynaptic, it also takes a bit of time. So an example given here is a knee-jerk reflex. The time is about 12 to 19 milliseconds, and the central delay time is 0.6 to 0.4 milliseconds. So this is an example of monosynaptic. So the central delay is not much because it's just one synapse. When you have bisynaptic, it will be greater than 0.7. Maybe it will be 0.14, 0.15 seconds. That's just the central delay time, depending on the number of synapses. So the more the central delay, it means that you have more synapses, or you are involving more of interneurons or relay neurons in the spinal cord. So this is a property of reflexes. The third one, recruitment and irradiation of stimulus. So mild pain stimulus, minimal contraction of flexor muscles and inhibition of extensor muscles. So if you have a mild pain stimulus, you find that you just have minimal contraction. So not all the muscle fibers are going to contract, are going to be involved in that flexion of the limbs or inhibition of the extensor muscles, because the pain is mild. But if it's stronger, you find that you're going to recruit more motor neurons so that you have more muscle fibers contracting at the same time. So even the force of friction or the force of withdrawal from that strong stimulus to be greater. So spreading of signals, radiating of the stimulus, and involvement of more or more motor neurons. This is called recruitment. So if you are recruiting more motor neurons, even the force of contraction is going to increase. You know that. So or may irradiate to other limb in case of crossed extensor response. An example whereby you're stepping on a nail and you know that is very sharp. So of course, you're going to cause friction of the limb or the leg to move away from that sharp object. And then the information is going to irradiate to the next segment so that you have extension of the other leg to support your weight. That we call the limb crossed extensor response. So you find that the stimulus will radiate to the other segment. So it's called intersegmental. So the increase in the number of active neurons is called recruitment of motor units, so that you have an increase in force of contraction, or you're increasing the force of response. You're going to get a stronger response depending on the number of motor units you are recruiting. Then the other one, the fourth one, is the after discharge. So a weak stimulus, but adequate, will cause a quick friction response. So this friction response, it's quick because you are responding to a stimulus which is adequate, even if it's weak. But a strong stimulus leads to prolonged response. So a strong stimulus will result into prolonged response. So the prolonged and repeated firing of motor neuron, which is called the after discharge. So the after discharge is whereby you've removed the stimulus, but some of the interneurons, they are still firing, or they were slow to fire the action potential. So you find that even if the stimulus is not there, but you're still getting a response. So that response is called the after discharge. So after a reflex contraction, if the stimulus discontinued, muscle relaxes gradually, not at the same time, because you have some interneurons that are still firing. So you find that you're still getting a response even if you've removed the stimulus. So the mechanism is due to the interneuron go on discharging, and also impulse takes longer time to reach the muscle through the interneurons. So you're going to have the after discharge. For instance, if there's something that is biting you, you'll be able to remove. So there'll be a reflex that will be able to remove that insect that is maybe biting you. But in as much as you've removed it, you will still get a response that is responding to that, even if the stimulus has been removed. So that is the effect of after discharge. So you can see in this diagram here, you apply the stimulus on the receptor. And then you have latency. This is just the time it takes for action potentials to move to the spinal cord and then back so that you get a response. Then you can see that you have a warm-up where you have this response that you are getting. Then if you remove the stimulus, so the stimulus is off here. Even if you remove the stimulus, but you're still getting a response, this is called the after discharge. For instance, when you're looking at this dog, it's scratching. Maybe there is a flea that is moving on the skin or biting on the skin. The dog can use the leg to remove that, but it will still continue scratching that because of the after discharge. There are still some interneurons that are firing an action potential. So you're still getting that scratching response even when the stimulus has been removed from the skin. That is called the after discharge. Then the fifth property is the final common pathway. So the final common pathway is simple. You're just looking at the alpha motor neuron. So the alpha motor neuron is the common pathway for the reflex. So the magnitude of response to stimulus depend upon the balance between excitatory and inhibitory input of the alpha motor neuron. So whether you're having excitatory or inhibitory, but these excitatory and inhibitory, they are still communicating with the final pathway of the reflex, which is called the alpha motor neuron that is innervating the extrafusal fibers. So inputs of the alpha motor neuron, same spinal segment, and interneurons from other segments, descending pathways from the brain. So therefore, alpha motor neuron act as the common pathway for activity in the skeletal muscle. So for you to have that response of the skeletal muscle, you would need the stimulation of the alpha motor neuron. So the alpha motor neuron is the final common pathway for these reflexes that involves the skeletal muscles. So you can see the inputs that are coming here. Some of them are stimulatory, others are inhibitory. So you can have stimulatory and excitatory postsynaptic potentials or inhibitory excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So others are inhibiting, others are stimulatory. So depending on whether you're having more of stimulatory than inhibition, then you can stimulate the motor neuron, then you get a response. If you're having more of inhibition, then you can't get a response. But the common pathway is the alpha motor neuron that is innervating the extrafusal muscle fibers. Going to number six, central excitatory and inhibitory state. Central excitatory and inhibitory state. Temporary changes in excitability due to subliminal fringe effect, which results in spread of impulse in spinal cord. So prolonged changes due to reverberating, so the prolonged changes due to reverberating circuits or neuromodulators that are involved. So in simple terms here, just looking at inhibitory or excitatory state of the spinal cord, whether you're having the central excitability or central inhibitory. So the central excitability, the excitatory influence is greater than the inhibitory influence. So if you have more of excitatory than inhibitory, then you're going to have central excitability. So you're going to excite the spinal cord, for instance. So after spinal cord injury, sometimes you can have a mass reflex as example of central excitability. So the central excitability, you find that the motor part of the spinal cord is stimulated, and also you can stimulate the autonomic nervous part because of the central excitability that can be due to some injuries. Then you find that you have generalized response to a stimulus. That will bring about mass reflex. When central excitatory state is marked, the excitatory impulses irradiate not only to many somatic areas of the spinal cord, but also to autonomic areas. For example, in chronic paraplegias, mild noxious stimulus cause, in addition to withdrawal response in all the four limbs, you find that the patient can also be stimulated in terms of urination, defecation, sweating, blood pressure fluctuations because of central excitability. So this central excitability will stimulate the motor part of the spinal cord and also the autonomic part of the spinal cord. You are having a stimulus, a mild noxious stimulus that can also stimulate urination, defecation, sweating, blood pressure fluctuations because of central excitability. Then central inhibitory, it's a state in which the inhibitory influence is greater than the excitatory. So if you're having more of inhibitory than excitatory, then you're going to have inhibitory or central inhibitory. Then the last properties or the last property of reflex is habituation and sensitization. Habituation and sensitization, these are two opposite things. So we have habituation and sensitization. So modification of stereotyped response by experience. So in this case, if the stimulus is benign, maybe just localized, you find that the response will decline. It will start declining. This is as a result of habituation. So I'll give you an example. For instance, if you put something in your pocket and that thing is pressing your gluteal maximus muscle. So initially you feel discomfort because of that stimulus that is pressing against your gluteus maximus muscle. But with time, you find that that response will start declining, and then that discomfort will go away. So it's more like you are just becoming maybe insensitive to that stimulus to some extent. Initially, it can be a discomfort, but later on, you find that you intend to ignore that stimulus, and then you just become comfortable again. That is called habituation. So prolonged stimulus can result into habituation, whereby the response will start declining. But sometimes you have noxious stimulation that will result into prolonged facilitation. An example, it will bring about sensitization for hours to days. So an example I can give here is, for instance, if you bend your finger, one finger, then after a day, you put your fingers in warm water. Warm water is not supposed to to stimulate you to feel that pain. But sometimes you find that the finger which is bent is going to be sensitized to the extent that the warm water will feel like pain, and then you get the pain sensation that is not supposed to be there. So this is an example of sensitization because of prolonged noxious stimulus. So a typical example I can give is allodynia. If you remember, allodynia is whereby a stimulus which is not painful is perceived as pain. Like the example I've given you to say warm water feels like it's hot water, but it's just warm. Why? Is because there is hypersensitivity in a certain particular area of your skin as a result of injury. Okay, so that is called allodynia, whereby you have a stimulus which is not painful, but is perceived as painful. Then sometimes you can also have hypersensitization that will result into hyperalgesia. Hyperalgesia is whereby a pain stimuli is perceived as more painful. So something that is bearable, for instance, it's painful but bearable, but because of hypersensitization, it will result into perceiving that stimuli to be more painful. So it's going to be increased in terms of the response. So these are properties of reflexes. So if a question comes, can you explain properties of reflexes? You should be able to do that. Okay, so with that information, thank you very much. So we are done with reflexes. So in the lab, we'll do the reflexes. So we'll be able to do the deep reflexes, then some of the special sense reflexes or cranial nerve reflexes. So all that we'll do in the lab. Okay, thank you very much for your time. And then next time we meet, we'll look at lecture 3 in motor system. So the lecture 3, maybe we'll just discuss the autonomic nervous system and the cranial nerves before we start going deep into the motor system. So once we have this background, then we are ready to start looking at the corticospinal tract or the motor system in general. Thank you very much for your time.